Okay, so good afternoon. Just calm down. So it's a pleasure for me to introduce you our speaker for today. So this is Jordan Cuff from the University of Newcastle, who's going to talk about something I hope very interesting. We will see in a few minutes. So just a short introduction to Jordan. So he did all his studies basically from bachelor to master to PhD at the Cardiff University in the UK. And he has been studying or mainly interested in the arthropods, in the biology of arthropods, and mainly the trophic interactions. And he's been using modern methods, such as molecular methods, to identify the interactions with the metabarcoding. And later, or actually his topic of the PhD was, I have it here, nutrient-specific foraging and the role of biocontrol, spiders as biocontrol agents. His main focus is on spiders, but he doesn't work only with spiders, but basically any insects. And I think, as you can see in these pictures, there are also aphids and coccinodebitals and parasitoids, so it's not going to be only about spiders. After the PhD, which he has finished in 2020, that means 4 years ago, he still worked as a postdoc for 2 years at the Cardiff University, and he worked on different topics. And one of them, for example, which was news to me, that you have monitored the COVID virus in the water. So using the molecular methods, of course. And then from 2023, he's got a new position at the Newcastle University, which is in the northeast of England. And that's where he's now. He's got an academic position there. He's mainly interested, as I said before, in the interaction between arthropods, using different types of methods, from molecular, but also, let's say, new powerful methods from robotics or monitoring using cameras and so on. And he's trying to implement the interactions with the nutrient foraging, and he's actually a lead for the foraging ecology group at your department. Yeah. So I'm very much looking forward to this topic, so the stage is yours.
Thanks so much, Sterling. That's great. Yeah, so I'll be taking you through a bit of a roller coaster ride today. So the main topic will be integrating these nutritional data into ecological networks, but we'll go through a few other processes along the way. So Stano's stolen my thunder, so I don't need to talk too much about this slide here. But just to kind of summarize that I've done everything from earthworms in my undergrad, tree rock hole communities in my masters.
How could you ever abandon that?
I know. I'm so sorry. And then my PhD was mostly on spiders, and I've done everything from wastewater to oilseed rape insects along the way. So that's all brought me to starting my own lab group. So for the last year, I've been building my group called the Foraging Ecology Research Group. And the main focus of our research is looking at the determinants and consequences of trophic interactions. So that could be the constraints, so the things that stop trophic interactions happening, the drivers, so the things that cause them to happen, the feedbacks, so the things that essentially perpetuate those interactions, and the buffers that essentially stop any of the bad consequences. And I'm going to be focusing mostly today on nutrients as a driver of trophic interactions. But along the way, we'll be talking about everything from weather and competition to prey availability and disturbance as well. So I'm gonna start off by just talking about spiders as biocontrol agents briefly, and how they essentially take part in these large ecological networks. And then we'll go on to talk a little bit about dietary DNA metabarcoding. So that's using DNA to look at the diet of animals. And then we'll go on to talk about the biocontrol and prey choice and nutritional dynamics of spiders in field systems, before then finally coming on to the ecological network analysis. So I thought I'd start by just talking about the importance of spiders. So many of you may be aware that they do a great job for us in agricultural fields. So they're known for eating our aphids. Here's a really nice figure from Michalko et al. 2019. Many of you hopefully have met Radek Michalko at some point, or maybe you will. But essentially, this is really nice evidence showing the role of spiders in suppressing pests. So we can actually see that it's not just the density of spiders, but also the diversity that increases that predation of pests, or the reduction of their density in farms. So clearly they have an important role. But actually there are other papers, such as Simonson et al. 2002, that suggest that maybe this control is very inconsistent, and essentially we need to try and optimize it. And in order to optimize the number of pests that are eaten by these spiders, we first need to understand what's actually going on. So this is a really simple ecological network. So you can see we have the nodes, which are the circles, they're the different species in the network, and we have the link, which is what essentially shows that they're interacting together. So this is a perfect example of biocontrol, but actually there's a lot more usually going on. These spiders are distracted by alternative prey as well, so they're not just eating the pests. And they're also in direct competition with other predators. So it's not just a simple thing of them getting the food, they have to compete with and sometimes even fight these other predators. And they're all feeding on their own alternative prey as well, and then they're eating one another, and there are parasitoids parasitizing them. So it's a really complex menagerie of interactions. So to really get to the basis of how this is happening and why this is happening and when it's happening, we need to start understanding the wider context of where it's happening as well. And just coming back to that simple example of spiders eating pests and other predators, we can see that there's a lot going on between the spiders. That's intragill predation. They're eating lacewings as well, which are other predators of crop pests. So not all of the interactions they're engaging with are actually beneficial. Some of them are detrimental. And some of them as well will be influenced by things like the abundance of the pests. So if we have loads of thrips, we might expect this spider to be eating more thrips than some of the other things. But one of the other mechanisms that's likely to be driving these interactions is nutrients. So this is a really nice figure from Mainz et al. 2005. And essentially in this study, they showed that when invertebrate predators are reared on a protein-rich diet and then given a choice between protein-rich and lipid-rich prey, they'll go for the lipid-rich, and vice versa as well. So from this, we can basically see that these invertebrate predators are selectively balancing their nutrient intake, much like many of us try to do on a daily basis. But most of the evidence for this kind of research is done in lab studies where we have sometimes quite simplistic and sometimes unrealistic systems as well. So in this case, we have a carabid beetle feeding on mealworms, flies, and I think that's a bark louse. But actually, in a natural system, much like that network I showed you earlier, there are a whole host more species, so it gets more complicated. So actually, trying to align these results together can be quite difficult. So ideally, we need to start taking some of these studies done in the lab into the field. And to do that, we need to know a few different things. So if this is the menu of things available to the spider, much like us when we go out for lunch, it can choose any of these different prey. But you can see that the nutrient values differ between them and the availability differs as well. So for us to really start to understand these nutritional dynamics, we need to understand that availability of the prey, the nutrient contents of the prey, and also, most importantly, what the spider's actually eating. So to address that last point, what the spider's eating, I'll talk through a little bit of the methods that we use to determine that. So traditionally, the most effective way of working out what a spider's eating in the field is to observe it. And I think that's still very much true today. If we see something happening, we know it's happened most of the time. If you get philosophical, we'll stray away from that. But to actually look at what a spider's eaten afterwards, it's a little bit more complicated. Because it might be that we can only look at these spiders in the field during the daytime. They might be interacting with loads of different prey at night when we can't see them. Or they might be doing these interactions in secret locations, like under leaf litter. So to address that in mammals and other vertebrates, people tend to use hard parts analysis. So it's where you go through the feces and you pick out the bits of remains of the different prey. So you can see there's a leg of some kind of fly, I think. And then I think that's a bit of a mouse jaw. So you could start to identify what these animals have eaten. But with spiders, they're fluid feeders. So what you get out is far less identifiable. And we also can't look at the predation of things like earthworms that are soft-bodied using these hard parts analyses. So essentially we need alternative methods. And there are a whole load of different methods available. So this is from a paper that Sano and I published earlier this year. It's basically a guide to help us decide what methods to use. So some of the questions we might ask are, Are there specific contexts in which the interactions are important? So are we looking for a very specific time or place? And for us to understand the nutritional dynamics of a predator, that would probably be a no. Is the time frame and specific identity of the prey important? Yes, it is. We need to know the nutrient contents. Are the interactions well characterized, common and predictable? In this kind of system where we haven't necessarily studied comprehensively what these spiders are eating, and because they're incredibly generalist predators, so they eat loads of different things, that would be a no. And do the focal interactions comprise a small set of known prey? And again, we don't necessarily know what these spiders are always eating, so it's a no. And that comes on to high throughput sequencing. So when something eats something else, obviously it ingests the body. That's one of the core parts of eating, actually ingesting it. And that means within its guts, it's got all of the bits of those prey. And that includes the DNA. So there are little bits of DNA floating around in the guts of the spiders that we can actually then use to identify what it's eaten. And the same can be said as well for parasitoids. So if they're parasitizing aphids, the body of the aphid contains the DNA of the parasitoid. So we can start to build these interaction networks just looking at the molecules. And this relies on a principle called DNA barcoding. So much like when we scan a barcode in a shop to find out what bottle of orange juice we're holding, we can look at the sequence and the DNA to identify what kind of fly it is. So doing this, we can take even really morphologically similar species, such as many parasitoid wasps that are quite hard to distinguish just based on morphological identification. And we can sequence their DNA and find out which species they are. And we can do this for whole communities in parallel. So just from one sample of a spider's gut or anything else really, we can start to construct whole communities of organisms. And this has been a massive boon for ecology. So we can take samples of water or soil, even air these days. You can filter the air and take the DNA and find out what's nearby. But also feces, as I just mentioned. And we can construct these communities on massive spatio-temporal scales. And obviously the most important example of this is the guts of spiders, because that will help us answer our questions in this talk. So when a spider eats the aphid, the process of actually finding out what it's eaten is extracting the DNA from the guts. So we mash up the spider and we essentially separate the DNA from all of the proteins and the things that we're not interested in. And then we sequence the DNA, and essentially we can then identify that the spider is eating the aphid. But there's a fundamental process in the middle of that, which is polymerase chain reaction. And raise your hands if you've come across polymerase chain reaction or PCR. There we go, most of you. I think COVID has really helped with that. It had to help with something. But we've all done PCR tests at some point, whether we know it or not, basically. And this PCR process essentially involves taking the DNA, heating it up loads so that it splits into two separate strands, and then we cool it back down again, and then these PCR primers that are designed to match the DNA clip on, and then we heat it up a little bit again, and then this polymerase enzyme fills in the blanks. So we essentially end up with a nice copy of the DNA that we're looking at in that specific region that we're targeting. So it means that we go from one strand of DNA to many in a very short space of time. And with all of this heating and cooling, it's just like baking a cake, a really tiny, transparent cake. And we have special PCR primers that we design to do this kind of thing. So you can see we've got all of these squares that are yellow and green and red and blue, and that represents the variation in essentially the gene of the species that we're looking at. So these are loads of different spider prey. So each row is a different taxon. And then we have spiders at the bottom as well. And you can see where it's yellow here, except for that tiny bit of blue, we have the yellow G here. We'll ignore the black letters for now, but you can see we've got the T's where it's green, the A's where it's red. So we can design these PCR primers to follow this sequence that should match all of these different prey. And this is great. It means that we can start to make copies of the DNA. But for a spider, it's a little bit more complicated because it's not just the prey DNA that we'll get. This is a very rough cartoon of what a spider's gut sort of looks like. So it goes in through the head, and then sometimes it diverts into the legs. Who knows why that's a good idea? And then it goes into the abdomen, and it branches out, and it just creates this mess. So essentially, we can't dissect out the guts. We just have essentially one big walking gut. And it means when we do this PCR process, the vast majority of the DNA we get is actually that of the predator itself, so the spider. And most of the time we know what spider we're looking at. So you can see here, some of them have close to 100% spider DNA that we're detecting. And the prey DNA is far lower. So in that case, we need to start to think about how we can isolate just the prey DNA so we get more data on what we're actually interested in. And this is a figure from a paper we published last year that just goes through different types of dietary DNA samples. So with fecal DNA, which I mentioned earlier, there's less predator DNA because it's just the feces, it's not coming from the live body of the predator. But the prey DNA is also very degraded because it's been through the entire digestive process. And if we flush the stomachs of an animal, which is common in birds, for example, we can get a similar effect where we isolate the gut contents from the actual animal itself. So we can reduce the predator DNA, but we can sometimes have quite a strong digestive effect on the prey DNA as well. So one of the most effective ways of doing it is just this whole body DNA extraction, which is our only option with spiders, basically, because we can't separate the gut or its contents from the spider itself, which sadly means that we get this really strong signal of predator DNA. So coming back to this, if we can't separate the gut from the spider and we can't separate the prey DNA from the predator, our best option instead is to try and exclude the predator DNA. So we can redesign those PCR primers to match the prey but not the spider. And when we exclude the spider DNA from the PCR, we end up getting much better results. So you can see there are still some problems, and we still do get some spider DNA coming through, but we can start to improve the data we get back. And one of the most interesting things we found doing this is actually that the communities of prey that we detect are really different between the two PCR primer pairs that we used. So if we just amplify a bit of everything, for example with this one here, the points are basically further apart if they're more dissimilar, so each point represents the diet of one spider detected with one PCR primer, and the red ones are the PCR primers that amplify everything, including the spiders, and the blue ones just excluding the spider from the PCR. And where they're joined by a line, it's the same sample. So we can see that we actually detect different diets by using these two approaches. So actually one approach doesn't tell us everything. We need to essentially use both together, which complicates things and makes them a little bit more expensive. It gives us better data. So one of the other problems with this approach is that it's really sensitive. If we, for example, have a parasitoid parasitizing an aphid, and then the beetle comes along and eats that aphid, it's not just ingesting the DNA of the aphid anymore, it's ingesting the DNA of the parasitoid. So when we put that through the sequencer, we'll essentially assume that this beetle is eating both the parasitoid and the aphid, which can sometimes be a bit confusing because it's not actually, the parasitoid didn't factor into the foraging by that beetle. And this becomes even more complicated when we start to look at omnivores. So here we have a lizard that's eating these caterpillars that are eating these leaves. And the lizard isn't eating any plants at all on that particular day, it's just eating caterpillars because they're nice and juicy. So when the caterpillar's going through the gut, it's got the plant DNA in its gut as well. And we have to use different PCR primers to target the DNA of the animals and the plants. So essentially we end up detecting the same two things in exactly the same proportion. So we're assuming that this lizard eats just as many leaves as it does caterpillars, which can be quite misinformative. And as well, we can't integrate context into these data. So if we have this little raccoon eating an apple that's covered in flies and ants, we'll again assume that it's going to eat the flies and ants. It might be targeting ant nests or something. So we basically need to be careful about how we interpret these kind of data. And this is particularly important when it comes to merging methods together. So if we're observing things in the field and we're separately taking the spiders and analysing their gut contents, we essentially need to be careful about how we treat those data together. I just want to apologise for the eye icon as well, because when I first made this figure, they only had an eye with conjunctivitis. But they have since released a normal eye, and I forgot to replace it. So when it comes to merging these data types together, we have all these different considerations that we've just talked through to think about. So we have the PCR primer biases, whether it's missing certain groups or amplifying everything. Obviously with our direct observations, we have biases as well. So we need to make sure that those biases don't cause problems for one another. We also have a lack of quantification with these molecular data. We do get quantitative data back, but it's sometimes inaccurate. We also have completely different perspectives of sampling. We have false positives where we're detecting things like those secondary consumptions, also lab contamination, and there are a whole host of other issues as well. And just to touch on one of these problems, to give an example of how we might start to tackle these, the primer biases can mean that whilst this is what the wasp's eating in a natural system, this is what we might detect if we have PCR primers that don't target Hemiptera. So we're missing the aphid and the leafhopper there. And that means that we're actually missing the indirect interactions between the wasp and or at least one of the 4 plants. So if we use multiple primer pairs, like we talked about with the predators, then we can start to maybe fill in those gaps and essentially create more complete ecological networks. So just to conclude on this section, we can clearly accurately identify species based on their DNA. Hopefully that's been a compelling enough argument for that. We can also identify whole communities from tiny traces of DNA from even the air. And we can therefore identify these interactions by looking at the DNA in the guts, and that we also need to be quite conscious of these obstacles and challenges when it comes to interpreting the data. So now we'll come on to some actual data. So I'm going to be talking a bit more about spiders as biocontrol agents, like I introduced at the start, but using some data that we collected a couple of years ago. So obviously coming back to this figure, we have not just the spiders eating the prey, the pests, we also have them eating one another and the fellow predators. So we have this intragill predation and biocontrol happening in parallel. So to start to explore these kind of questions, I went out to some farms in Cardiff in the south of Wales in the UK. Anyone who's been to Wales will know that this isn't a very good representation. This is a really lovely day, but it's raining like 90% of the time, which is really challenging for fieldwork actually. But I was going around collecting these money spiders from these fields of barley, and at the same time I was collecting wolf spiders as well. And whilst I was collecting the spiders, I used this vacuum sampler, which you can see here in first person perspective. It's a bit like Call of Duty or something. It's the entomological version. But I was going around collecting the insects with my vacuum sampler to find out what prey were available to the spiders in each of the locations we were taking them from. And then, much like in the first part of the talk, we were using DNA metabarcoding, so taking the DNA from the spiders and sequencing it to find out what they'd eaten. And when we compared the number of pest taxa that we detected, so things like aphids and thrips, versus the number of predators that they were eating, so particularly one another. Spiders seem to really love eating one another. We can see some really interesting differences between the different spider genera that we looked at. So we looked at 4 different money spider genera, so Linyphiidae, and then we also looked at, where is it? There we go, Pardhosa, which is a lycosid, so a wolf spider. And we basically found that the diversity of pests was much greater than the diversity of predators in the guts of most of them, but particularly Tenuophantes and Bathyphantes ate loads of pests. But you can see here that the purple bar for the predators is really squidged down for Bathyphantes. So they were eating actually far fewer predators than Tenuophantes. So the biocontrol potential of these spiders differs between different spider genera. And when we look at the life stages, it's really interesting as well. So we can see that the adult spiders are eating quite a few pests, but also quite a few predators. Meanwhile, the juveniles are eating more pests and fewer predators. So basically, what this means is if we want biocontrol to be effective, we want the nice young spiders to be coming up through. They're doing a good job. But this doesn't tell us everything. We don't know what the actual foraging decisions are of the spiders. We don't necessarily know what their preferences are. We just know what they're interacting with. which could just be driven by what's available to them. So we can start to investigate these questions. So much like we can take the spiders and do sequencing to work out what they're eating, we can take the prey community data that we collected from our vacuum sampling and use null modeling, which is basically where we model what we expect to happen, to create another network where we have a spider eating things based on how available they are. And we can then compare our observed interactions from the metabarcoding with what we expect to happen based on the prey availability. And this allows us to start looking at the selectivity of spiders in the field. So just to give you a bit of an example of what that might look like before we look at the actual data, if we have loads of thrips, a few aphids and a single fly, we might expect that the spider's eating loads of thrips and a few aphids and probably missing the fly altogether. But if we then take the DNA data and we find that actually it's eating thrips and flies, then we can see that maybe it's eating as many thrips as we'd expect, but it clearly seems to be seeking out the flies in amongst the mixed community and potentially avoiding the aphids, considering it's not eating any of them at all, despite them being quite abundant. So we can start to look at that in ecological networks. So this is a bit complicated. I'm not expecting anyone to take much away from this. But essentially the blue lines are where they're avoiding something. So you can see most of these avoidances are for thrips. And they're particularly wide because there were just so many of them. There were just loads of thrips all the time. But you can see these red lines as well, which are where the spiders are eating things more than we'd expect based on their abundance. So they're preferentially seeking out some things like aphids, for example, which were not that abundant in the fields we were looking at. And we can start to use this to look at some really interesting contexts. One context that I find really interesting in agriculture is harvest. So here we have that, again, really nice sunny field of barley. This is probably a little bit more like the sky in South Wales. But you can see it's exactly the same field, but it's just been completely harvested. So we just have this stubble left over. So I liken this to a mass deforestation event, because we go from having this really complex vegetation structure to having just bare ground overnight, basically. And so we explored some of the reasons why this is important in a paper we published earlier this year with Stano and Radek and others. So essentially, we have these really different processes going between the transition of different periods of the year. So in the early season, we have immigration of biocontrol agents, and then coming on to the primary crop growth period, particularly in early summer, we start to see intra-guild interactions taking place alongside suppression of the pests. And then when this harvest comes along, we're expecting to see a lot of prey switching, so where predators are changing what they eat because the prey community completely changes, and also a lot of emigration that's driving that. So we essentially looked at the prey communities, pre and post harvest, and the spider diets as well. And what we can see here is that the pre-harvest spider diets are distinctly separate from these post-harvest. There is some overlap. So again, the ends of these spindles are different spiders' diets, and how far apart they are is how similar they are. So the only thing that you really need to take away is that the pre and post-harvest blobs are separate. And when we start to look at the prey selectivity or preferences within these two periods as well, we start to see some interesting things. So pre-harvest, thrips are eaten about as much as we'd expect, which is denoted by the little white circle. But post-harvest, they're actually avoided. And there are many other taxa that change as well. So we can see this preference for springtails and flies pre-harvest that disappear post-harvest. But one of the things that's really consistent is that they seem to be preferentially seeking out one another. So Linyphiidae are always top of the bill. So they're clearly having a great time. So what about the weather? So we've looked at a management context, but what other processes are changing what spiders eat and what they prefer to eat? So I mentioned the really rainy weather in South Wales. It can be very variable, so it can go between these two almost seasons within a day, which can be very frustrating. But these again are plots representing the diets of different spiders. So each dot is a different diet, and essentially all you need to see is that the contours have different sort of progressions, so different shapes and forms. And essentially, which day of the year it is, the length of those days, and weather variables like wind, pressure, temperature, rain, and dew point all very differently interact with the diets of spiders. So there are essentially completely different relationships in all of these different weather categories. But when we change this to look at the prey communities of the spider, you can probably see that a lot of the patterns are actually quite similar shapes. So with the day, we have that sort of sweeping left to right. With some of the other ones, like pressure, it's also converging in the middle. So we can see that the prey community changes are actually somewhat linked to these dietary changes. But again, we can use this null modelling to look at the preferences of spiders. So each of these bits at the top is a different weather category. So hot is quite self-explanatory. That was the really hot days. But we have like dry and windy, high pressure, wet, low dew point, and wet, mid dew point. And essentially the preferences of the spiders differ depending on the weather, which is likely to be partly due to the prey community changes, but also partly down to different temperatures and other conditions. The two sort of main highlights for me were that when it's rainy, the spiders tend to avoid springtails, possibly because they're all coming out of the ground. And when it's nice and hot, the spiders seem to really love eating aphids, which good for them. I mean, it's quite a nice juicy snack on a hot day. But of course, I've talked about nutrients at the start, so it's time that we start to integrate those nutritional data into these networks as well. So in 2021, we published a method in Methods in Ecology and Evolution, which is basically for the extraction and determination of proteins, lipids, and carbohydrates in parallel from a single insect. So if we took, for example, a little parasitoid wasp, we essentially do all of these mechanical and chemical processes to end up with these nice colourful plates. And this all looks a little bit complicated, but essentially all it is, again, we can do our Call of Duty entomology to collect our insects. We then just smash them into pieces so that they look a bit like a soup. And then we essentially take bits of that soup and do biochemical reactions to work out the proteins, the carbohydrates, and the lipids present in the bodies of these insects. And we can then use those data to start to integrate it into our interaction data to look at how nutrition is driving that foraging. So importantly, I mentioned at the start about the nutrient-specific foraging behaviour, so spiders and other invertebrate predators seeking out the nutrients they need. So I mean, this is just a cartoon of a food label from the UK where I've replaced it with aphid values. I don't actually know what the percent of a spider's recommended daily allowance is, but if anyone does know, please tell me. But once we get these nutritional data, we essentially have all of those three pieces of the puzzle that we needed to explore these nutritional dynamics in the field. So from the spiders, we have the dietary data, so we know what the spiders are eating. From the prey communities, we have their availability, and we can start to look at that selectivity in the field. And from these macronutrient assays, we then have the nutritional data as well. So we have those three parts of the menu that we wanted. And when we integrate all of these together, we can start to look at some interesting processes. So here we have a ternary plot. So if a point's closer to the top, it's higher in carbohydrate. If it's closer to the bottom left, it's higher in lipid. And if it's closer to the bottom right, it's higher in protein. And each of these tiny points is the average nutrient content of the prey consumed by a single spider. And the big points are the averages for each of the groups. So you can see again, we've got these 5 different genera. And I think the most important point from this plot is that you can see the yellow is squidged down at the bottom. So we can see that the Pardosa wolf spiders are eating diets that are richer in protein particularly, but also some lipid. But they don't tend to be eating much carbohydrate at all. Meanwhile, the 4 different money spider genera are stretching slightly higher up into the carbohydrates. This is all in a paper that I've put the QR code there for because I'm testing fate, because I finished the proofs on the way over here, so it's going to be published any day. But if that QR code doesn't work and you do want to read it, just let me know. But we can also look at other characteristics of the spiders, so we can look at the sex of the spiders, so you can see male and female. They overlap pretty well. There are actually differences between their nutritional intake or the nutritional content of the prey that they eat, but it doesn't necessarily come out too well in these plots. And the same is true for life stage as well. So you can see the adults and juveniles eat quite overlapping prey nutrient contents. But again, we come back to that question of preference. So we know what they're eating, but is that just driven by the nutrients available to them? So we can use those null models again to work out what we think the spiders will be eating based on what's available to them. And we can work out the average nutrient content of the diets that we expect the spiders to be eating and compare those to what we actually see. So you can see the greenish blob in the middle. That's the nutrient contents of what we expect the spiders to eat, and the purple is what they're actually eating. So we can see that the spiders are eating nutrients in different proportions to what we'd expect, and they stretch in all three directions. So different spiders are individually eating imbalanced nutrient intake. And when we come back to that question of nutrient-specific foraging, one interpretation of this might be that this is a spider that's just had a heavy day eating lots of carbohydrates. It happens to us all sometimes. And maybe tomorrow it'll go for a bit more lipid or protein, but this might just be single snapshot points within that sequential foraging of the spiders for different nutrients. And we can represent the actual individual groups of spiders as well within that prey choice context. But each of these points in these ternary plots represents a different prey taxon. So this one might be aphids, that one might be thrips. I'm completely guessing. But the important point here is that you can see there's a red dot near the top in all four of those ones. So the Bathyphantes, Origone, Microlinophia, and Tenuophantes, but not for the Pardosa. And what this essentially tells us is that these four money spider genera are preferentially seeking out at least one carbohydrate-rich prey taxon, whereas the wolf spiders don't seem to. And what's quite interesting is that the position of that point changes. So they're essentially differentiating which carbohydrate-rich taxon they're targeting. Meanwhile, the wolf spiders seem to really love their protein, denoted by the big red blobs at the bottom right. And again, we can look at differences between the sexes and life stages. So males absolutely love some carbohydrates, whereas females don't seem to. Adults, again, quite heavy on the carbs, but not so much the juveniles. So we can start to predict and understand these interactions from these kind of analyses. So there are a few different questions left over. So we don't necessarily know how these interactions with the prey containing different nutrient contents affect one another. And from the network that we looked at the start, we know that it's important to investigate how those indirect interactions also take place. We also don't really know how the nutrients are structuring the food webs more generally. We just know that they're sort of driving some interactions. And we don't necessarily know how that might lead to the indirect driver of biocontrol. So that's when we really come on to the main topic, which is integrating these nutrients into ecological networks. So if you're wondering what this might tell us, networks essentially allow us to look at things at the community level. So rather than just looking at one interaction, we get an idea of what's going on across the whole ecosystem. So we can start to look at how these nutrients are structuring food webs, and we can start to look at how that might be driving ecosystem services like biocontrol as well. So this has been done before in some ways. So I think my favourite example is this one on the left from Timberlake et al. In 2022. So essentially they looked at a plant pollinator interaction network. So we have different pollinators here, so honeybees, flies, wasps, you name it. And then up here we have the different crops that were present in this place. It's essentially a small community in Nepal. And we can see the frequency of interactions between these pollinators and the crops. And then they've taken it a step further by looking at the vitamins and micronutrients that these crops provide to the local people. And this is amazing because it essentially allows us to see the value of these pollinators in providing nutrients to the people through this indirect interaction. And there are other examples as well, like this one from Senior et al. 2016, which looked at other nutrients in that sort of network context as well. So I wanted to set out to do something similar. So this was my first attempt at doing it, which is in retrospect terrible. But essentially this is the network you saw earlier, but instead of having different taxa at the bottom, we have different groups that are created by clustering the nutrient contents together. So this group here might have thrips in it, but it might also have aphids if they're a similar nutrient content. But we can see that the carbohydrate-rich taxa are over here. We have a few lipid-rich taxa in the middle. So we can start to look at the structuring of the interactions, not based on the Linnaean taxonomy, but based on how the nutrients are distributed across taxa. So this is essentially how the clustering process works. So we can see that there are different coloured blobs there, which say which taxa were grouped together, and it's all based on similarity of nutrient content. But this was quite limited because it didn't necessarily tell us anything about the nutrients within the interactions. It was more as an end point of the interactions. So instead we can take an approach a bit like that one of the plant pollinator community in Nepal. So we can actually represent the nutrients as nodes in the network. So each of these green points is a different spider, and we can see how or a different spider group, and we can see how they're interacting with the different nutrients. And we can even start to look at just the average intake of spiders by comparing the nodes across the whole network. We can also represent nutrients as whole networks. So rather than looking at the 3 different nutrients, we can look at the exchange of nutrients between spiders and their prey in terms of carbohydrates, and then lipids, and then proteins. And this is essentially the direction that we ended up going down. So if we take the ecological network that we've constructed, we have the consumer nodes, so in this case that's the spiders at the top. We have the resource nodes, so that's the prey that they're eating. And then we have these interactions, so the links. And the thickness of the links essentially tells us how frequent those interactions are. So we can start to look at the mathematical properties of the network by investigating those links. But if we multiply those links by the nutrient contents of these resources, we can essentially get the transfer of nutrients from prey to predator. So we can start to characterize that in the network. And then we end up with something like this, where we can look at the resource nutrient content at the bottom. We have the 3 different links for each interaction, telling us about the carbohydrates, the lipids, and the proteins. And then we can even represent the mean intake of nutrients by the spiders. And this is what that looks like in practice. It looks a little bit like something you'd need 3D glasses to look at properly. I promise you don't. It doesn't get any better. The key thing isn't necessarily the visualization of the networks, which almost always look a bit chaotic. The real benefit is when we start to actually explore the mathematics behind the network. So we can start to look at different properties. So just to explain these graphs, you can see the yellow points for the carbohydrates, the blue are for the lipids. Oh, what's happened there? The computer didn't like it. Oh, there we go, we're back. That was just a moment of suspense for everyone. And then we have the red points that are the proteins, and we have these black lines, and that's where we've done another null network, where we've redistributed the nutrient contents randomly. So rather than an aphid having high carbohydrate content, it's just swapped its nutrient value with loads of other things. And we do that 1,000 times so that the nutrients are randomly distributed across the network, so we can see what the network might look like if nutrients just don't matter at all. So the black lines are essentially represent the variance of those 1,000 simulations. So when we see a point that's quite far from that line, we can see that there's quite an interesting effect going on. So weighted nestedness is essentially where we have interactions of some nodes in the network that are subsets of the interactions of others. So it might be that one spider's just eating 2 things that another spider's eating alongside another 2 things. So it's that nesting of interactions within one another. And what we see is lipid is quite nested. So there's a subset of lipid interactions within wider interactions of other consumers. I think the really interesting ones, though, are modularity and the other ones to the right. So modularity is a measure of how much you could just split the network up. So if you just got a knife and cut down the middle, would you be able to do that without cutting any of the links? So does it form distinct modules? And what we see is that carbohydrate is really low, so much lower than we'd expect, which shows that it's actually really widespread across the network, far more so than the other nutrients. Everything's getting its carbohydrate from all over the place. And we can also see the same for H2, which is essentially a measure of specialism, or speciality, specialization, that's the one. So we can see that specialization is very low for carbohydrate, again suggesting all the spiders are just getting carbohydrates a little bit from everything. And we can see the same again for linkage density, which shows us that essentially the density of links, so the number of interactions going on, is quite low generally. So there's not much carbohydrate, and it's being taken from a whole range of different resources, which I thought was interesting enough to ask some more questions. So we can then split the prey, or split the networks rather, so that it's all of the interactions, so what we've just been looking at, but we can also look at just the beneficial prey of the spiders, so when they're eating other predators or other taxa that are beneficial, like pollinators. And we can also look at their interactions with pests, and we can compare the network properties between these groups. And what we see is that the beneficials, there's quite low generality, which again is just a measure of how many interactions the spiders are engaged in, or how much of each nutrient they're taking. But the pests, you can see that we've got lower generality for carbohydrate and much higher for protein and lipid. When we look at the vulnerability, which is the number of interactions from the prey, so how much nutrient is being taken from each prey taxon, we can see again quite a bit lower for the beneficials. And for the pests, you can see a lot of lipid is coming from the pests. So the pests are quite lipid rich, or at least their exchange of nutrients to the predators is biased toward lipids. And we can look at the modularity again, and we can see that the modularity is particularly low in the pests for the lipids, which means that essentially the lipids are being taken quite evenly from all of the pests. And a question that made me think about was competition, because it implies that most of the spiders are actually competing for the same pest species or taxa, and essentially targeting them for lipids. And this is where networks get really interesting, because we can start to look at competition through the lens of networks. So this is the same network, but where these light grey nodes are each individual spider, and the dark grey nodes are the different prey taxa. And we can essentially look at competition by looking at when these two spiders, for example, are sharing this one prey. And we can start to draw links between them, so we can look at competition through the lens of the networks. And if we just isolate that to the competitive interactions, we can see these red links are where the spiders are directly competing with one another in the same location. And what we found so far from this short investigation, we're currently finishing this work at the moment, is that all three nutrients are really important for competition. So the spiders are essentially competing with one another, driven by the nutrients. So we can also look at various other things through the lens of nutritional networks. So I mentioned nutrient-specific foraging at the start. We could look at how the degree, so the number of interactions of each consumer changes, and that might reflect some nutrient-specific foraging. If, for example, generalist consumers are either eating just a bit of everything or targeting nutritionally complementary prey. We can also look at nutrient limitation in networks. So if there are very few carbohydrate-rich taxa, for example, we might expect that they're targeted by more generalist predators, because they're essentially targeting that one resource rich in carbohydrate. We can look at other things like generalism versus specialism, so whether specialists can survive on different nutritional properties to the generalists, and also new concepts like nutritional naivety. So when a species arrives at a new location, is it going to have to try and learn where the nutrients are, or does it innately know? And these are all questions we can begin to answer through this framework. So essentially, nutritional networks can tell us how nutrients are structuring interactions. They can help reveal some really poorly understood ecological dynamics, such as nutrient-specific foraging. And they can also allow us to explore how these nutrients indirectly drive interactions and how they drive indirect interactions. So the next steps for this framework are basically to develop more null-nutritional network approaches. So I talked about where we can randomize the nutrients between the different prey. We can also start to look at other kinds of null networks where we essentially change the nutrients within the network within controlled constraint. We can also start to look at how network rewiring rules depend on nutrients. So rewiring is where it's essentially prey switching within a network context. So it's where we see a predator change what it eats. And it might be that prey becomes unavailable or is otherwise removed. And at the moment, we just assume that they'll just move on to something else randomly. But of course, nutrients are super important for them choosing what to eat. So we can start to predict better what these predators will move on to. And we can also start to look at robustness of ecological networks from nutrient perspectives. So robustness of a network is essentially when we remove the prey nodes, how many predators lose all of their interactions. And that's referred to as a secondary extinction. So if we essentially remove all of the carbohydrate from a network, nothing should really survive. So we can start to look at that nutritional depletion, and that can help guide things like conservation and other sort of habitat management. So just to conclude the whole talk, we've seen that molecular methods provide unprecedented potential to study these natural systems. We can use these data to explore some really interesting things like nutritional ecology within a field context. We've also seen that nutrients dynamically affect the interactions of spiders and the ecosystem services they provide, like biocontrol, and also the ecosystem services that they're detracting from when they're eating some of the things that we like pollinators. But with this nutritional network concept, we can also start to study how these nutrients are structuring entire food webs and ecosystems. So the next steps from here, really, for me, we have a whole load of different projects starting in my group at the moment. Everything from artificial light and predator plant commensalisms through to ecoacoustics and projects focused on gene regulation. So hopefully next time you see me, which hopefully will happen again at some point, we'll be able to talk about some other really cool topics along these kind of lines. But thanks so much for listening, and thanks to all of the fantastic collaborators involved in all this work.
So thanks a lot for showing us the amazing results, and we have time for plenty of questions. So,
First of all, thank you very much for this very colorful talk. It's not only literally colorful, but also very interesting and very diverse. Some years ago, I don't know how much this has been going on, there was quite a buzz about, let's say, ecological stoichiometry, which, if I understood this correctly, boiled down to this saying of what you are, what you eat. So basically showing that organisms, that their composition of tissues would be very much affected by their diet. But on the other hand, of course, we all know, starting from ourselves, that we choose what we eat to a large extent. But sometimes we cannot choose so much. So how does these two things compare? I mean, on one hand, you have this specialization or these preferences. On the other hand, it has been shown that still there is a lot of underlying stuff which is being ingested anyway, and it does affect the tissues and their composition.
Yeah, so to touch on your first point first, the colourful figures that I've used, I've been reprimanded for in the past. So I actually got told to remove one figure from a paper because it was borderline psychedelic. That was the words they used. So I'm glad you can appreciate it.
Well, my professor told me when I studied that colored graphs would be unscientific.
Yeah.
But it was almost pre-computer times, you know.
Yeah,
so.
I'm happy to be unscientific if that's the case. But to touch on your main point, so there's a lot to be learned from ecological stoichiometry in this kind of context. And in fact, that's the field that's really been studying nutrients in an ecosystem-wide context already. So what we're doing is kind of taking more of a nutritional ecology approach to the same kind of questions. But in terms of the you are what you eat aspect, we did some preliminary work with fruit flies where we essentially fed them different diets and looked at differences in their nutrient contents, their cuticular hydrocarbons and their volatile organic compounds. And we found that all of that completely shifts. So we essentially see that it is true, you are what you eat. When we fed flies these lipid-rich diets, they became lipid-rich, and essentially they smelled lipid-rich, and they tasted lipid-rich. So it's a really interesting point, because obviously what we have in the networks I've shown is quite a coarse representation of things, where we're assuming that the taxa have consistent nutritional content, and to some degree that's true. There'll be constraints to the variation with which they change their nutrient contents, but they will change, and the predators can theoretically sense that if there is a sensory difference in those nutrient contents. So really what we need to move toward is taking a more individualistic approach and looking at these kind of networks where we know all of the individual traits, like the nutrient content of the individual prey, but of course the spider's already eaten it by that point, so it gets really hard. But it's, yeah, fantastic point.
Okay. Yes?
For how long the prey's DNA stays in the spider's body? Or how many plus spider's meal can you track?
What was the last part, sorry?
How many spider's meals can you track?
Yeah, good questions. So the how many is difficult because the quantification of the data is really tricky. So what we essentially get from the sequencer is the number of reads or copies of the DNA that the sequencer's read. So that can relate to how much of the prey was eaten, or it can relate to how recently it was eaten. And we can never really know which one is true. So there are some really interesting studies going on in Austria at the moment where they've used RNA instead of DNA. And RNA is really sensitive, so it basically breaks down much quicker. So by comparing the DNA and the RNA, we can maybe start to look at how recent those detections are. But in terms of how long the DNA stays in the gut, there are loads of different studies. There was a really nice systematic review on that topic from, I think it was nearly 20 years ago now. But they essentially showed that we can detect DNA for sometimes up to 2 weeks or even further, sometimes a month. But when we're using this metabarcoding approach, there's a bias toward the most abundant DNA, and essentially we're not going to detect things that have really degraded and have been in there for a long time when there's more recent meals to detect, which will definitely outcompete the data in that PCR process. So it's hard to say with certainty. It's also impacted by things like temperature and other sort of metabolic effects. So yeah, that's probably the most vague answer I can give you, for which I apologise, but that's as far as we know really.
I have another question.
Yes.
When you are making that spider soup from which you extract the DNA, it is from individual spiders or?
Yeah, so we just use the abdomens of spiders, and then we can separate the DNA from everything else using a variety of different methods. In this case, we used silica spin columns, so it's where essentially we press the DNA through centrifugal force through a filter, and the DNA gets stuck to the filter and everything else goes through. But nowadays we're starting to use magnetic beads that pull the DNA out of the soup, and then we can get rid of the liquid and keep the DNA and purify it that way. It's
done from a single spider?
Yeah, a single spider, just one single abdomen.
Even when they're very small?
Yeah, so even the juvenile spiders, so the juvenile money spiders are sometimes just a millimetre or two, and we can still get good DNA from their gut contents.
Another question? Yes.
You showed that the juveniles are better for pest control. Is there some way to exploit it, or is it just useful information?
Yeah, I don't know. We could open like an old spider's home on the edge of the farm. Yeah, I'm not really sure how we can do that. But essentially just making sure that there's high population turnover is pretty key. How we achieve that is another question altogether, but I suppose having a diversity of predators within these systems will likely keep the populations in check, and the older spiders are less likely to be sort of strong competitors anyway, presumably. So yeah, that's probably the best we can do for now, at least.
Any question? So I would like to comment when you guys ask about you are what you eat, and you mentioned the experiments with Drosophila, that actually when you fed them with let's say lipid-rich diet, they smell like lipids and so on. Because this is actually answering the question what David Mines and Soren Toft published in the Science paper 20 years ago, when they actually showed that actually predators like carabids and spiders are able to select prey on the nutritional composition, and they didn't know how.
Yeah.
They observed they can select it, like if they were lipid short, they would select the prey which is lipid rich, but they didn't know how. So
Yeah.
now we actually, so did you publish these results?
No.
Ah, okay.
We basically had very few replicates, and it was quite a short. It was a pilot study to do something more afterwards, but then COVID came and everything kind of got stalled. But the other problem we had was that the lipid-rich flies didn't like being very lipid-rich, so they preferred dying. In fact,
But they still,
yeah.
but they still smell lipid-like, right?
Yeah.
So did you analyze the cuticular hydrocarbons? Did you
Yeah.
do that? Okay.
Yeah, I can show you all the data afterwards because
Yeah.
it's in my thesis in the last chapter as a I wish I'd done more of this point.
That would be really interesting
Yeah.
to look at, because I mean this is answering the question why it happens. I mean, how can
Yeah.
they actually select?
Yeah, so we tried to get funding for a study where we'd look at the electroenterogram responses of beetles to different nutrient intake as well. And we couldn't get the funding, but that's definitely something I'd love to do at some point.
Yeah, sure.
Because, again, like you say, it sort of answers that fundamental question.
Right. And then I have another question. When you were analyzing the nutritional composition, you focus only on the macronutrients,
Yeah.
carbohydrates, proteins, and lipids. How about the micronutrients? Did you look at it as well or not?
No, so we didn't look at any of the micronutrients, but they're super important. There's so much evidence that different amino acids can drive foraging, for example. So it's not just about the protein, it's about the kind of protein and the composition of it as well. So what we have here is quite a coarse look at quite a complex system, but to be able to do it at this kind of scale, we essentially need to start slightly coarser and zoom in as we go.
So could you look at the same sample size again? You look focusing at the micronutrients?
Definitely, yeah. And with the technologies we have now, it's becoming more and more possible. So we can start to build more complex networks. And just flicking back to the nutritional network we looked at here, like this is just 3 different link types, but actually there's no limit to what we could do. It might look terrible, but we can use the sort of mathematical principles of network analysis to look at this in an almost limitless number of dimensions, which is a very exciting part of it.
Okay, another question? Yes?
Now I want to poke a bit. You are in the ecological department, right?
Yeah.
No agricultural or forestry stuff, right?
So my building's called the Agriculture Building.
Okay, that might explain it. We also in the zoological seminar here. So is it okay to actually look through all this through the prism of beneficial and pest species? I mean, from a purely ecological point of view, these are very anthropogenic points of view which do assign some value to species which as scientists we should kind of reject and be very neutral on that.
Yeah, I'm completely with you. So the main analysis that we did here was completely irrespective of any sort of pest or beneficial status. But we followed it up with that analysis because essentially one of the main questions that we got the funding for was to explore that agricultural context.
I thought there would be a funding thing.
Yes. And I think, there's an interesting question there. But of course, if the beneficials and the pests have the same nutritional content, then it doesn't really matter to the spider. I don't think the spider's going along and thinking, oh, it will help those humans out if I eat this one instead of that one. But obviously for us, there's an important consequence in our agricultural productivity.
Sure. You have a question?
Yes, I also wanted to thank you for a fascinating talk. I really enjoyed it. And my question was related to the one that's been already asked. So if there's a way how to find out if when exploring the diet structure using the DNA sequencing, if the results can be somehow biased by the different rates of DNA degradation within the gut, or if somebody explored if when we feed the spider something, if the percentage of what we sequence from the gut will be equal to the percentage.
Yes, there are loads of really good studies. In fact, there's a fantastic meta-analysis. It's Lam et al. From 2019. And they basically looked at the quantitative potential of metabarcoding in a whole load of different studies. And I think the most fascinating part of it is that the results are really ambiguous. So some studies, there's a really strong link between the biomass that's actually being consumed and the number of DNA reads that we detect. And in other studies, there's almost no relationship at all. And the best part of that is the citations of that paper are from studies that are using it as evidence both for and against the whole concept. So
This is for spiders or other organisms?
all organisms, yeah, or yeah, at least a large range of them. So a lot of the research has been done on like marine mammals, like seals, but also things like bats. That tend to be more common groups.
But for spiders, you believe it?
I don't tend to use the data quantitatively because I'm just always a little bit skeptical about how reliable it is. And instead we can use binary analyses, so we can, all of our models use a binary data structure. So in that case we just have a presence or absence. When it comes to the networks, of course you can see the spider groups have different thicknesses of line, and that's because we have multiple individuals per group, so we can look at things as a frequency of interaction within a group rather than a frequency of an interaction within an individual. And then if we look at networks like this one, this is all just single interactions, so you can see that they're all the same width, basically. But if we look at the competitive ones, again, it's then the frequency of those competitive interactions between individuals. Does that make sense?
Okay.
Cool.
Okay, another question or comment? If you can go back to the slide, I want to have another question. Yes, no, this one, yes, this
Yeah.
one. Can you tell me how many pests are included here and which specific pests are included?
It's a good question. I couldn't tell you off the top of my head.
I assume the aphids.
Yes, so it'll be
And then the
aphids,
thrips.
thrips, there were some Lepidoptera.
This was on the cereals, right? So
Yeah.
It's wheat or what is it?
It's
Oh,
barley.
Barley.
Yeah.
Okay, so maybe the flies. What
Yeah.
you call it?
Frit flies.
Not fruit flies.
No, frits were
Argeo
new.
is no Argeo.
So chloropids.
Chloropids.
Yeah.
Chloropid flies.
Yeah.
So because all of them are feeding on the same basic food, that
Yeah.
means the barley. That's the fact. So basically, also the composition of the pest should be very similar.
Yeah.
That's what I would assume.
Yeah.
So did you find it that actually the composition of aphids would be very similar as that one of the thrips, for example?
So this figure is probably one of the best ones to show it because each of these points is a different prey taxa. And you can see that there are clusters. Like here we have quite a few taxa that are a bit protein rich, but mostly very rich in lipids. Whereas here the carbohydrate rich prey are much more distributed. But none of them were like identical. We just have this sort of similarity in some groups. But it's a really good question actually. We have a student at the moment who's doing nutritional analysis of loads of different taxa we collected from a wheat field with the idea of looking at differences between taxa and between time points as well. So maybe that would be a really interesting question to explore around the pests and whether by feeding on the crops they end up with more similar nutritional profiles.
Yeah, so then for the predator it wouldn't really matter which kind of pest it's eating because let's say the composition of the aphid is the same as that of the thrip.
Yeah.
Probably not exactly the same but very similar.
Yeah, so at that point it becomes more about the ecology than the nutrients. So the capture efficiency and all sorts of other things. But yeah, it's a really good point. Yeah, amazing.
Okay, is there any other question? Yes?
It was a nice, straightforward talk.
Thank you.
Maybe my question wouldn't be that much straightforward.
Okay.
I'm thinking how to put it in, to explain it, but there was one thing that grabbed my attention, and it was like, okay, there's a field, crop field, you have pests, and then you have spiders that are competing for that pest, meaning that there are no pests. So you know, you don't want to have the pest, so if they are competing, there's no pest, but we always have some pests, so it was, is there really competing, meaning that the pest is limited resource?
Yeah, I mean, it's a great question, and I think it's something that we need to factor into those analyses of competition, because if the prey are really abundant, then that competition no longer really matters that much. But it's not something we've explored just yet. But the other part of this is that all the spiders that were competing were within the same location. So although it may be a location that's swarming with thrips, for example, they're both eating thrips within that same place. And what we see in the literature is that spiders will often co-locate with large abundances of prey. So sometimes they'll put their webs in optimal locations where the prey are most abundant. And they can even change the sort of structure of their webs to reflect different prey types. So it may be that if they're occupying the same space, eating the same prey, their webs will be in sort of physical conflict, if nothing else. But yeah, it's a really good question.
Thank you. And another one was about in the system that you have one system which you have really nicely studied from different angles. So my question is if you try to plan to do some choice experiment, because being a spider, I would definitely select aphids against thrips. They are tiny, lots of cuticle, and try to avoid it if I have another choice to go for.
Yeah, so do you mean sort of lab choice
Yeah,
experiments?
a lab choice.
Yeah.
So if you push spiders because they're just flooded by thrips, okay, you know, I'm hungry, so just give me some thrips. But if you offer them the aphids, you'll just avoid them. And then we'll, of course, change your result based on what is available and what they are actually preferring.
Yeah, so I was actually talking to Stano about this yesterday, because we did do some choice experiments, but I have an amazing ability to just kill everything.
Okay,
So-
I'm on the same page.
Yeah,
Maybe.
so I dropped a tube with a spider in my car boot once, and I didn't notice it for 3 months. And then I opened the tube and the spider got out completely fine. But when I'm trying to look after them in the lab, they just die. I don't know. It's clearly me, I must be the problem. But we did do these experiments looking at their choice preferences for, it was fruit flies, aphids, and springtails. And we did get some information from that, but essentially there was no real strong effect. My suspicion is that it's actually just to do with the motion of the different prey. The fruit fly was comparatively quite large, quite a lot more mobile, and probably came across the spider more, whereas the aphid, very easy to catch, but it was the other side of the dish, walking really slowly, checking each grain of charcoal it was on. So I think the results were quite a lot impacted by that. So actually that was part of the reason why we did the fruit fly experiment, to see whether we could get one individual prey item that we could feed the spiders that differed in its nutrient content, but had the same morphology and activity. But we essentially couldn't find a reasonable enough way to differentiate between them. So the way that we'd love to go with that is actually looking at the signals of the prey without actually putting the prey in there. So whether that's vibrational cues or semiochemicals or something else. Yeah, thanks so much.
Okay, another question? I have another one.
Yeah.
I'm actually wondering, do you think that the preferences in nutritional consumption would change over season? And actually, the first I would like to know, how long did you run the study? And was it in spring, which I would assume, or late or early summer?
Yeah, so it's sort of spring, early summer. So it ran for a few months. But it's, yeah, fantastic question, because the developmental stages of the spiders, the weather, all of these things are likely to impact it. But because of the fact that we didn't take the prey nutrient contents across that time period consistently, we didn't necessarily have confidence that we could test that within this context. But it's something that we definitely want to move toward, because if we could actually look at those nutritional differences in the prey and in the diets of the predators across that timescale, I'm sure we'd see some really interesting signals.
Yeah.
Yeah.
There's a lot of work to be done.
Yeah, definitely.
You're at the beginning of your career, so you...
I think it's all mapped out for me now. I'll need a couple of careers.
Yeah, well, everybody was...
Many students.
Yeah.
So, any other question? Okay.
Maybe this is irrelevant, but I was just curious, how did you create the pictures of the spiders? Was it some AI-driven software? Or?
So, pictures like these ones were all made in BioRender.
Yes.
But before I had BioRender, I used to draw them in Microsoft Paint. And I was telling Stano earlier, because I saw Stano's incredible drawings. And my drawing of a spider recently, I was asked if it was an aphid. So I clearly didn't have it. I do not have any artistic talent, so this is all BioRender.
Did you use any AI in your presentation?
No, not at all. Yeah, maybe I'm missing a trick there.
Another question?
Yes, okay.
This is also irrelevant, but like, when we are talking about the pictures, I noticed in one of the first diagrams there was a dolphin.
Yeah,
As a prey.
I did this in a conference recently, and I thought that no one would notice. And as soon as I put it up on the screen, everyone burst into laughter.
Oh, really?
And I thought, wow, I thought it was really secret. But it was my little Easter egg to see how much attention people were
I
paying. There you
waited
go.
if someone else would ask, or if I'm just.
Oh yeah.
Okay.
Yeah,
You got me.
I was putting together the network, and I was thinking, what else would this beetle eat?
Yes.
And the only thing I found was something that wouldn't even occur in the same country as the beetle. And I thought, well, at this point, it may as well be a dolphin. And I thought, well, why not? So there we go.
It's living on the beach and there is some dead dolphin on the
Yeah,
Could be.
beach.
there we go. Yeah, good spot.
I was wondering if I'm just stupid and everyone knows that.
I never heard of a dolphin-eating beetle. My goodness.
Brilliant question.
So, is there any other question? If not, then I'd like